Cade Mirchandani, a PhD student in biomolecular engineering at UC Santa Cruz. My lab studies symbiosis, the long-term partnerships between bacteria and animal hosts. Today I'm going to tell you about a project where we measured something that's been really hard to pin down. How many symbiotic bacteria actually get passed from a parent to its offspring, and we're going to look at this in a deep sea clam system. You've probably heard of mitochondria, the powerhouse of the cell. What you might not know is that mitochondria were once free-living bacteria that became trapped inside our cells over 1 billion years ago. That process, endosymbiosis, is one of the most important events in the history of life. We study modern versions of these partnerships to understand how they work and how they evolve. In the deep sea, there are clams that host bacteria in their gills, bacteria that feed them. These clams can't eat on their own. They're completely dependent, just like we depend on our mitochondria. But there's a cost to this dependence. At each generation, when a mother clam lays eggs, only a small number of bacteria make it onto those eggs. That's called a bottleneck, and it means that the symbiont population loses genetic diversity every generation. Over time, harmful mutations build up irreversibly. This is something called Muller's ratchet, and it slowly degrades the symbiont's genome. The question is, how severe is this bottleneck? People have tried to answer this by counting cells under a microscope, but that only tells you how many cells there are, not how many genetically are distinct from one another. So we took a different approach. We used 2 ultra-accurate DNA sequencing methods. Each one corrects its own errors in a different way and applied them to the same tissue from a single clam. By looking for rare genetic variants that both methods detect independently, we identified 20 high-confidence mutations in the symbiont population. Then we built a computer simulation of how symbiont populations evolve through repeated bottlenecks and used machine learning to figure out which bottleneck size best explains the pattern of mutations we observe. What we found is that the effective bottleneck is about 8 symbionts per generation. That's it. Just 8 genetically distinct bacteria making it through to the next host. Previous estimates from counting cells suggested hundreds or even thousands. Our number is orders of magnitude smaller. And yet, despite this incredibly small bottleneck, the symbiont genome is still surprisingly large and intact. That tells us something important. There must be some other force counteracting the degradation. We think it's occasional horizontal transmission, where symbionts from other clams mix in and restore the genetic diversity. So, why does this matter beyond deep sea clams? The same forces that are degrading the symbiont's genomes are the same forces that shaped mitochondrial genomes over 1 billion years ago. By studying modern symbioses like this one, we get a window into one of the most fundamental transitions in the history of life. How free-living bacteria became the organelles that power every cell in your body. Thank you.